Thank you. And I'm here today to talk to you about a study that we have been doing at NIH for quite some time, for about 3 years now. And it sort of addresses a lot of the points that were brought up in the discussion today. This is a large study. We are trying to describe neurodevelopment among a normative set of children. We plan to share all of the data as quickly as possible as the study is going along, and we are hoping that you all will make use of it. And that's really my main point. My main reason for being here is to make you aware of this study and to make you aware of it as an opportunity for you. So the Adolescent Brain Cognitive Development Study is a longitudinal study of about 10,000 children starting at ages 9 or 10 and following them through early adulthood to assess a number of different factors that affect brain development and functional outcomes. So why are we doing this study? As we all know, adolescence is a time of extraordinary development, physical, emotional, and intellectual. And we still don't know a lot about how childhood experiences affect this sort of development and affect different types of outcomes, including academic achievement. Why now? When we decided to do this study, one of the things that we were very concerned with, so I'm from the National Institute on Drug Abuse, and this is now a multi-NIH institute study, but one of the things that we were most concerned about was the fact that the laws were changing in the United States. Marijuana was becoming legal in many states. Children were starting to use vaping devices, which is actually now just really ballooned up to about a third of high school students, and they're using products that contain nicotine. We don't even really know that much about what nicotine does to brain development. So all of these things were happening, and even in situations where we did tend to have an idea where we could say that children who start early and use marijuana frequently have a whole variety of adverse outcomes, we were always plagued with the question of how do you know these children weren't different to begin with? And we really didn't know. So our thinking was we should study a cohort of children before they start using substances and then see how exposure to substances and a whole variety of other environmental factors affects their brain development. At the same time, the technology was advancing sufficiently. We could do imaging studies in multiple labs and get comparable data. The Human Connectome Project sort of started this and developed methods that allow us to get very precise measurements and, again, do them across multiple labs. And big data was also becoming a very important an important area that I think we're going to, that obviously we still need to learn a lot about how to maximally use big data, and I think that the ABCD study is going to be pushing this and hopefully pushing our ability to improve the technologies and to improve our analytics for using big data to answer questions that we're all very interested in. So these were the original research objectives that we thought about when we were designing this study. And again, some of these are things that you just heard about. We wanted to be able to describe individual developmental trajectories and what kinds of factors could affect them. We were interested in developing national standards of healthy brain development. We are very interested in investigating the roles and the interactions of genes and the environment. We are collecting genetic information, and also among our cohort we have about 2,000 multiple births, mostly twins. So we will be able to also look at the influence of genes and environment through that particular lens. We were interested in a whole variety of different factors that could affect development, and that includes physical activity, sleep, screen time, sports injuries, relationships with parents and peers, where you live, and what the sort of neighborhood factors are that could all affect the overall development of a person. We were interested in, this is also a time when we often see the onset of mental illness, and so we could begin to look at the factors that influence that, as well as how early would we be able to detect certain types of mental illnesses. And we've always been interested, again, in mental health and substance use. And again, we have this problem of what's causing what, or is there an association, or does exposure to one worsen the course of another? So this is another area that was important to us, as well as how the use of different substances affect each other and affect developmental outcomes and sort of gateway theories that people have been proposing for many years. So we have a total of 21 sites in the US. This is kind of standard. There's always that kind of big gap in the middle. Obviously, we needed to have research sites that had the capability of doing brain imaging and that had a lot of different types of expertise, psychological expertise, computational expertise, the ability to do longitudinal studies and to be able to recruit children and keep families engaged in the study for a long period of time. So we developed a cohort with these 21 sites that are using a single protocol at all of these different sites. We are recruiting from schools because most 9 and 10-year-olds are in schools, and it was thought that that would probably be a pretty effective way to get a good representative sample. And we also, as I said, have a number of twins. So we have 4 sites that are bringing in a mostly twin cohort, although we also have twins at some of the other sites. And now we have about 2,100 kids that are enrolled that are twins or multiple births. We have enrolled 11,874 children, families. This has been in a 2-year, 1-month period, so we had a pretty aggressive timeline, and our researchers met this timeline, which is utterly amazing. I'll give you a sense of what the cohort looks like in terms of the demographics. We tried to, as best we could, match what the census is like in the US. So this is the American Community Survey. It's done every year, and we use it to give us an idea of race, ethnicity, as well as other factors that I'll show you in a second. In our singletons, we were pretty close to matching the American Community Survey. For some reason in our study, we have greater numbers of people who have mixed race, and I don't know if that's just that people are now identifying themselves that way more often, or if this is really something that is changing. With our twins and multiple births, we were a little farther from matching the overall community survey numbers. That's not unexpected because a lot of the twins and multiple births follow fertility treatments, and that tends to be in higher SES people, and it tends to be in families that are more likely to be Caucasian. Another area where we did struggle, we tried, especially at the end, to make a big effort, but we have also a little bit of a skewed sample in terms of highest household education, and we're skewed on the side of having more education. And this is, again, not surprising because we are recruiting from areas around these research sites. I will say that the catchment area for each research site, if you look at the overall catchment area for all of the research sites, it covers about 25% of the population of 9 and 10-year-olds. And each site was given a recruitment objective that was based on the demographics of their area, so that they could be at least, we could get, as best we could get an epidemiologically informed sample and a diverse sample, although it's not nationally representative because everyone does not have an equal chance of being a participant in this study. But we wanted to make it a study that was as diverse as possible. We have very few exclusionary criteria, so we basically brought in anybody that was willing to come and participate. This is the baseline protocol. I know you're not going to be able to read all of it. I wanted to give you a sense of just how intensive it is. This is a 7-hour protocol. It includes about 2 hours of imaging, measures among the youth of physical health. We collect biospecimens. Mostly we were collecting saliva samples from the 9 and 10-year-olds. The twins were also collecting blood samples. We also got hair, and we got baby teeth. We have a number of different measures of mental health, neurocognition, culture and environment, and this is also very important. Among other things, we're looking at discrimination and acculturation, which we think can be very important factors in affecting neurodevelopment, and of course substance use, which was what our institute in particular was very interested in to begin with. We also use other more passive data sources, so we do get geocoding from their residential history. We are trying to get school records. We would like to get electronic health records. We're still sort of figuring out how to get that. But we are trying to use these other data sources to be able to provide additional information that we're not getting necessarily directly from the participants. Again, the parent protocol was also quite extensive. Most of it was asking about their child, but we also got information about the parents' physical health, about their use of substances during pregnancy, about their mental health. And in addition to that, we asked them a number of questions about their children. So this is the general timeline of the study. It doesn't cover the whole study, but every other year we have these very long, intense assessments in which we also do neuroimaging. We collect blood samples or saliva samples, biospecimens. We do all of these variety of assessments, and we talk to both the parents and the children. In-between year, we do bring them in also for a fairly comprehensive assessment, but we don't do neuroimaging. And what we've been doing, and I'll show you this in a second, is as we keep developing the protocols as we go along, so some of these are going to be consistent from year to year because this is a longitudinal study, and that was, of the most important aspect of this for us. But at the same time, even though a 7-hour protocol sounds like a lot, there were a lot of compromises made, and there was information that we couldn't necessarily get at baseline, so we tried to get some of that information at the 1-year time point, or we will get them at other time points. Some of it also has to do with the maturity of the child and what is appropriate to ask about. We have, the 2 years later will be the same sort of neuroimaging as well as all of these different assessments. We also do very brief phone calls in the interim period. Right now we're doing it at about 6 months. As they start to get older and start to use substances, we will try to do it every 3 months. The idea of doing these interim calls is both to allow us to pick up on early substance use or early changes in mental health. And again, these are 15-minute calls, just kind of getting a general sense of how the kids are doing. But the other point is that, because it is so critical to our study that we keep these children in the study, we try to have as much contact with them as possible. We have newsletters and birthday cards, and these calls are sort of part of our ongoing efforts to develop relationships with the families and to make sure that they want to be part of the study and make this something that they see as valuable. This is the 1-year follow-up, and I just wanted to highlight that there were a few things that we added into the survey because, as I said, we couldn't ask them the first time. One of the things we asked about was gender identity. This was something that the CDC, the Centers for Disease Control and Prevention, was very interested in, and they are now a partner with us. The National Institute of Justice is as well, and the National Endowment for the Arts. There are also other, we also started to get urine specimens from the youth in order to be able to look mostly at measures of, I think cotinine is what we were looking for there. So we are, the blood samples are mostly for genotyping. Oh, no, the urine samples, I'm sorry. The urine samples are, blood samples are mostly for genotyping. The urine is for that. The saliva samples we're also using to get measures of pubertal hormones so that we can assess exactly where they are in their puberty. And again, we added some questions to the parent protocol, and mostly these are just to sort of start to answer some of the questions that we were not able to include in the baseline. So I just wanted to, so what I can show you right now is just going to be some descriptive data because we only have some of this baseline data right now. The descriptive data that I'm showing you are from the first 4,500 children that were recruited. Those data have already been released to the public, and those are the data that both our ABCD scientists and any other scientists that want to work on this, want to work with these data are using for publication. So as I said, this is mostly descriptive since we don't yet have the longitudinal data to use to be asking the kinds of questions that we hope to ask. So just to give you a sense of the variety of types of information that we're getting, we are asking about extracurricular activities, how much time children spend in either participating in sports or other types of extracurricular activities. And we're also asking about their exposure to music and arts, and whether they get instruction, formal instruction, whether they're getting private instruction, or whether or not they're just learning these things on their own. And one of the things that's quite interesting is that if you look at these factors by parental education, you see that there is a difference, and that the higher the parental education, the more exposure to some of these factors you see. And we think it's important to be taking these sorts of things into account, because we're interested in very specific questions about how substance exposure may affect brain development, but there are many other factors in the environment that are going to also have an impact, and they need to be considered. What's also kind of interesting is if you look at sports, you see similar patterns, except with respect to football. And football is something that many people are really keeping their children from doing now because of all of the concerns about TBI. So I think that there's a lot of ways that we can look at this data that are going to tell us some hopefully important information about how youth are growing up. So we were hoping to get a substance-naive group at baseline, since that's one of the reasons we chose 9 and 10-year-olds. And in fact, it seems, at least in these first 4,500 kids, to be the case. What I'm showing you on this slide is we asked them if they've ever heard of these substances, and that was sort of where we started out. If they had heard of it, then we asked them follow-up questions. But as expected, most kids knew about alcohol and tobacco. Most of them knew about, 58% knew about marijuana. We asked about prescription drug misuse. We didn't use that particular terminology, and these were sort of less well known. We also included a substance on there that didn't exist, just to make sure that we weren't getting kids saying yes to things that they really didn't know what they were. And we got a very, very small percentage that said that they had heard of that substance. We're also able, because of the size, again, of the cohort, we're able to look at male and female differences, and males tended to have heard of more of these substances than females did at this age. So that is heard of, but when you ask actually if they have ever used or ever tried any of these substances, you can see that there's really very little use in this cohort. The bar on the left, which shows the most, is whether or not they've had a sip of alcohol. And there you've got about a quarter of the kids doing that. And we asked further whether or not that was done with their family as part of a religious ceremony, or whether they did it on their own without their parents' knowledge, since these obviously could have different implications in terms of risk. Almost none of them had had a full drink or reported smoking or using marijuana. Familial depression, this is another area that obviously everybody in this room, I think, is interested in. There were quite a number of kids that had some immediate family members that have a history of depression, so I think this is going to be quite interesting to see what happens as these children develop. If you look at the children themselves, they show pretty typical patterns. We have a little bit of a high representation of children with ADHD. We have about 8% of these kids have symptoms of ADHD. These are not clinical diagnoses, but the assessments are made using the KSADS, the Kitty Schedule for Affective Disorders and Schizophrenia. So it uses diagnostic criteria, but these are not diagnostic or clinical assessments. Nevertheless, this is what we see, and I think this is what, I mean, part of our, in looking at the baseline, there's a number of things that we're looking for. First of all, we're just interested in the descriptive nature of the findings with respect to our cohort, but also we want to make sure that it makes sense with what we know about normative samples from the population. We want to make sure that on some of the measures of neurocognition that you get the sort of variability that you would expect. So a lot of it for us was also validating that the measures that we were using were giving us the sorts of results that we would expect to see at this time point, and in fact we are. We also asked about suicidal ideation, and we asked this of both the parent and the child. This was a really tough one. Our research assistants were very uncomfortable asking about it. But the CDC has found that among 10 to 14-year-olds, suicide is the second leading cause of death. So it does happen in children these ages, and so we thought it was very important to be asking about it, and also to be asking both the child and the parent, because we wanted to see just how well these things matched. We got slightly higher values from the children than we did from the parents. If any of the children, in any case when the child did report any of the suicidal ideation, we have clinical people at each site, and there was always follow-up done just to make sure that this was not something that was an imminent problem, and if it was, then we have a protocol in place to tell the family and to tell the parents, and to also try to link them to services that they might need. One of our researchers, Deanna Barch, was very interested in psychosis proneness and whether or not you could detect at this age even some sort of likelihood of becoming psychotic. And so what she did was she modified a prodromal questionnaire that's used in adults and changed the language to be more understandable to children. So instead of asking, have you had experiences with telepathy, psychic forces, or fortune telling, she asked, do you feel like you've had special or unusual powers, like you could make things happen by magic, or that you could magically know what someone was thinking? And she found that there were a fair number of children that had some that had some indications of these sorts of behaviors. But she also asked the question about whether or not they were distressed by any of these phenomena. And of course, a much smaller group responded yes to that. When you look at the family history, those children that responded as having distress associated with these particular sorts of feelings tended to come from families where there was a greater likelihood of there being a psychotic disorder in the family, and not depression and not mania. So it does suggest that there was some selectivity, and that there may be something really valuable to this questionnaire that may inform us about future development of psychosis. She also looked at a number of the different neurocognitive tests, and the types of tests that she saw effects on are the types of functions that you see affected in people with schizophrenia. So she's trying to validate this measure, and I think we will only know with time whether or not it really is an effective measure and an early detection that could be used to help us identify children earlier on and potentially intervene earlier on. Screen time is something that everybody is very interested in, and so we did ask about various types of screen time, whether kids watch TV or movies, whether they watch videos, whether they play video games, and we looked at about how much time they spend doing that, and we also tried to compare weekdays and weekends. And the thing that I wanted just to point out here is the difference between girls and boys, which I guess, again, is not terribly surprising, but when it comes to playing video games, that tends to be something that is done more frequently in boys than girls. We also asked about social networking. And again, we have, right now, most of these kids did not have their own phone. That will change probably in the next visit that they have, but at this time they really didn't. So there wasn't a lot of use of social media, but there was some. And this time it tended to be a little bit higher in girls that would be texting as compared to boys. And there's already been a paper out on this, and the main point that I want to make about this paper, this is a paper by Martin Paulus, and what he found is that it's complicated. We keep getting asked the questions, is use of screen time bad for your kid? Is it bad for their brain? Does it change their brain? And what he found was that there were different patterns of use, and they were associated with different patterns of behavior and different patterns of brain connectivity. And an example was that if you look overall, individuals with greater screen media activity generally had less physical activity and greater sleep disturbances, but actually those who spent most of their time on social media had greater physical activity, better sleep, and less family conflict. So the message that we keep trying to give to people who are trying to sort of boil this down to one message or another is that it's not going to work that way. It's going to be nuanced. It's going to probably be changing over time. And this is an area that we are, extremely interested in, and we're really trying to figure out what's the best way to collect the most data in this area, and whether or not there are ways of using passive data collection, apps put on someone's phone to be able to measure screen time. So all of that we're sort of trying to pilot. But it's clearly an interesting area, and it's clearly going to be important when we look at relationship to mental illness, as well as substance use. But I think it's something that we need to really be cautious about how we talk about it. And very importantly, the whole approach to this ABCD study is an open science approach, and we want this to be a resource for the entire scientific community. So we have fast-track neuroimaging data. These are pretty much unprocessed neuroimaging data that is released almost on a real-time basis. There's probably about more than 9,000 images that are already available to the community. On the bottom there, that's a Lego version of an MRI, so that one of the kids did, which we thought was pretty interesting. But we've also done a curated data release of the first 4,500 participants, and we released that in February of 2018. And we expect to have the full baseline curated data released probably at the end of this month. I also want to mention that we have something called the Data Exploration and Analysis Portal, and which you can also get access to. And it was something that Wes Thompson and others from our data center created in order to make it easy for people that are not necessarily terribly experienced with these kinds of data to ask questions and to have sort of the right statistical models used based on what the ABCD data cohort is. And there's a lot of our data analysis and informatics center is trying to do a lot of outreach and dissemination of both how to use this particular analysis portal and how to access the data, because this is something that we obviously have no control over the data once it is released, but we really do want to encourage people to use it correctly, and we want to be there to help them figure out how to use it correctly. So this has been, this is one of the amazing things about this study, is that it's really been a culture change for the community. There's no embargo time in terms of the data release. There's really has to be a sense of, we are interested in all, what we're going to learn from this, but we're also creating a resource for the greater scientific community. And right now we have 7 publications out that have cited ABCD data. 3 of them are from ABCD scientists, 4 of them are from non-ABCD scientists. We also have a special issue of Developmental and Cognitive Neuroscience, which really describes sort of the whole process of how we came to do what we're doing in the ABCD study. So what NIH had in mind when we first launched this study, how the study is organized, how the different groups decided on the measures that they're using, what the role of the External Advisory Board is. So it's really sort of a primer on how to do that kind of a study if somebody wants to do that. But if you don't want to read it all, we're also really happy to talk to people about it and really share both the lessons learned and the knowledge and all of the procedures that we're doing so that others can use it as well. And I just want to mention another study which we're in the planning stages of right now. And again, it's very relevant for what everybody was talking about here. We decided to start at adolescence because we knew that the drug laws were changing in the country, and it was sort of urgent to know what was going to happen in adolescence, which is the main time that they start using drugs. But we also recognize that earlier development is equally critical, maybe more critical. And so we are now trying to do another large study similar to ABCD, but starting at birth. And the U.S. has a horrible opioid epidemic problem right now, which I'm sure you've all heard about, which includes a lot of children, or a lot of neonatal abstinence syndrome, a lot of fetal exposure to opioids. So this study is intended to both provide normative data on brain and cognitive development, but also to oversample mothers that are using opioids and other drugs as well. And we decided that this is an extraordinarily complicated study for many reasons, including many ethical reasons, particularly for women who are using substances and pregnant. So we are actually doing, right now we have March 25th is when our applications are due, but these are for planning grants. So we're having the group spend about a year and a half trying to plan what the study should look like and what are some of the factors that need to be taken into account so that this can be done in an ethical way. So we know that there's a lot of challenges just in doing, the imaging at that young age. So there's a lot of technical challenges, but they're really, at its core, if this is not a study that people feel, this study could really mess things up badly if it is really not done in an ethical way. And in the US, women they can lose their child, they can be arrested for using substances. So all of those things need to be taken into account, as well as a lot of the places that have these very high opioid use rates are places where there's a lot of poverty and there may not be a lot of access to services. So if we're enrolling people in a study, even if it's an observational study, we can't ignore the fact that you have people that are in need and that, because this could have very long-lasting effects. So trying to figure out what we can do and how to balance that without, at the same time, doing an interventional study is another area that we're hoping will be discussed in these planning grants. And we plan to have several meetings of the people that are funded, and we will make those meetings also publicly available so that people can hear the lessons learned. And then when the full cohort is funded, anybody is able to come in and apply for that. And so I'm ending here just to tell you who all of the different contributors are. This started out with NIDA and the National Institute of Alcohol Abuse and Alcoholism and the Cancer Institute. We were all focusing on some aspect of addiction, but then it expanded very rapidly to many other NIH institutes that all have very other important questions that they want to ask. And as well, we've brought in other agencies, including, as I've mentioned before, the Center for Disease Control, the National Science Foundation, and the National Endowment for Arts. And that's it.
Yeah, you should really be applauded for this, especially the way the data is being made kind of so open so quickly. It's really going to be amazing for the field. So really exciting. I can't wait to go back and have people. I'm curious about some of the. It wasn't exactly clear to me, but it could have been my attention. What percentage of the participants will have like genetic data of some sort, the SNP?
Yeah.
And then the second thing I was wondering about, it looks like you've done quite a bit to validate a lot of the kind of cognitive behavioral psychoeducational measures. I'm wondering about the medical history stuff. Is there like a small percent where you actually get the medical records or like get it validated so you can know how because I think the biomedical side of it will be also pretty informative with regard to brain health.
So we are collecting blood from everyone to be able to do genotyping and whole genome sequencing. Right now, we don't have the money to do all of the whole genome sequencing, so we're sort of still figuring that out. But the idea is that we do have the genotyping done on all of the participants, and we do hope to have that information. With respect to the medical history, I mean, we're asking a lot about the physical and medical history, but right now we don't have access to the electronic health records. One of the things that we are actually trying to do is to, we're thinking about it, we haven't gotten there yet, but the US also has this very large study called All of Us, which is the study that the NIH is supporting to try and get 1 million or more people across the country to participate in a research study where their data can be made accessible to researchers using electronic health records, and people can either self-select and become part of the study, or some of them are being selected through their healthcare systems. And what we were thinking about was actually trying to get the parents to join this All of Us study so that we could actually, because right now All of Us doesn't actually have children in it, but we thought if we could get the parents to join, then we might be able to get even more of their health information that way. So there's some issues that are complicated to figure out, like we need to make sure that we can identify the parents from ABCD that are doing this, but But that's one way that we could do it. I mean, we are trying to figure out what's the best way to be able to get verification of the health records.
Peter Jones.
Hello, Peter Jones from Cambridge. I'm also interested in the open data angle of this amazing study. And I see that you've taken a decision to make raw data available. And I wondered how raw it is. Say for imaging, have you taken faces off? And did you debate the pros and cons of making raw data available that people can get lost in for years?
Yeah, we decided to do that because there were people that were very interested in it and that might be interested more in developing methods that they could use the raw data to do. You know, we have, there's a data use agreement in which you have to say that you will not try to re-identify the subject and that you will also not share the data with anybody else. So we are depending upon the honor and of the people who are signing these data use agreements. But we you know, the Human Brain Connectome also made its data available, although they waited a little while, but their experience was that very few people used the raw data and almost everybody used the curated data. We really weren't sure how this was going to go for ABCD, so we just decided that we would do both, and we would hope that there would be, you know, that there would be, you know, that they would each have different purposes and that we could gain something from it. And I met somebody here earlier who is working with it, and who's working with the raw data and who's trying to use that to be able to identify individual trajectories relative to particular population.
Question back.
Hi, sorry, Kate Watkins from Oxford. Just first of all, a comment and then a question. So one of the things that we've just started is an imaging study here in Oxford of children with language developmental disorder, and we're using the protocols from ABCD.
Oh, great.
So this speaks to the question we had in the breakout sessions about pooling data. So I would encourage anyone else who's thinking about starting a study to think about using the exact same imaging parameters as are available to be able to take advantage of the bigger database and put their data in context. And then my question is, in my experience of these big kind of studies, we frequently encounter quite a large number of incidental findings in pediatric brains from presumed healthy participants. So I just wondered, do those data end up being available or are they taken out? And I mean, it's a whole other ethical issue, I realize that, but...
Yeah, I think that they're only taken out if they want to be taken out. We have all of the neuroimaging data read by a pediatric neurologist, and we have a rating scale for the severity, and obviously if there's a severe problem, then we will immediately contact the family and try to connect them with proper medical attention. I think that in terms of whether or not they stay in the data set, that is, I'm not positive, but I'm pretty sure that's up to them. If they're willing to stay in it, then we will. And we hope that actually this pediatric neuroimaging data can also be helpful in terms of developing norms and being able to get a sense of, you know, what is really outside of the norm. So, but at this point we do, I think we leave it up to the family whether or not they want to continue and whether or not they want their data to stay in.
Are there other questions? Yes.
Sarah Jane Bicknell from UCL. Thank you. That was such an interesting talk. I have a question about your characterization of social media and technology and phone and screen time. That changes a lot. You know, it evolves. Children
Yes.
a few years ago were on Facebook. Now they call it Granny Book.
I know.
They go anywhere near it, and they're on Instagram and Snapchat and presumably Applegate. How will you try to capture
Yeah.
content of what they're looking at?
So we have basically all of the decisions around ABCD are done by work groups. We have work groups on everything, including a work group on social media, and it's very tricky. I mean, we have done focus groups with kids just to get a sense of both the way in which they describe what they do as well as what are the social media platforms that they are using. But it's hard because it is changing so quickly. And right now we're interested in looking at the content, for example, of their Twitter feeds or but right now we haven't quite figured out how to do that. So we are trying to meet with different experts to sort of get an idea of what's the best way to do it. As I said, we're also piloting some apps that could be put on their phones to see if that will be able to identify, track their social media use, the kinds of social media that they're using. But we don't really know yet how well that's going to work. That's a really important challenge for the field. Everything is changing so rapidly, and this was, this was our first sort of foray into this, but we imagine that this is going to be more and more important as the children are getting older. So if you have some ideas, we would be really happy to hear them. And we are trying to reach out to different experts who are doing research in this area to get a better sense, but it is moving so quickly that it's hard.
Arnold, did you have a question?
Yeah, Arnold, who's not here from UCSF, may not have been paying careful enough attention, but I just wondered about the ethics and consent in particular, because these are children, when they start, they're young, and then, of course, they're going to get older. So I assume the consent initially is from the parents, and then at some point, the children will have some say?
Yes, initially it's a consent from the parents and assent from the children. And then as they get older, they will be switching to being consenting themselves. And we have a bioethics work group is one of our work groups, and we also have an OSMB, which is like a DSMB, but for an observational study. So their role is also to help us think through any of the ethical challenges that we are going to be faced with, and also to be reviewing the consents and also the burden on subjects.
Right, so the informed part of the consent
Yes.
Explains the potential risks of identification and the genomics and all the rest of it, I
Yes,
assume.
we do everything we can do to protect their confidentiality, but we do explain that there are risks. And we are using a single IRB for this, which is the UC San Diego IRB for most of the sites. And when this gets renewed, it will be for all of the sites because of the new NIH policy that requires it.
And last question.
It's a really interesting presentation, thank you. I just wondered, and thinking particularly about potential transatlantic differences, in terms of the sample, are you able to track prescribed psychotropic medication? Yes. And do you have a sense of whether the prevalence rates of, just thinking about your rates of ADHD, is the prevalence pretty representative of the US?
I have not looked at those data, and I haven't seen those data yet, but we are tracking that, of course, because it's obviously very important. And, and we've also been trying to talk with, with people from different countries that are doing other kinds of cohort studies I think everybody agrees that having this type of project being done in different cultures in different countries that have not only different just cultures, but different environmental factors that may be affecting it is really crucial. And there's a study in India that's looking at a variety of different places that have a variety of different environmental toxins, and there may be a study in China that will be kind of using these sorts of things. But I think that we are very interested in having this be this type of project and these particular measures be used as broadly as possible. And that will be an interesting point, because I'm sure there's a lot of differences in the psychotropic medications at different places.
We're going to have to end it there. Let's thank Dr. Weiss for a great talk.